Very good evening to all the participants who have joined already to this meeting. Special thanks to Professor Arvind sir, Professor Prakash Halami sir, Dr. Sharad sir, and all the participants who have come for this meeting. So today's topic is on microbial production of industrial enzymes. Uh, I think we have covered this similar topic related to enzymes, and on demand, this topic has been kept again because it is a very important topic from industrial viewpoint, as well as to show that how much important microbial systems are with respect to the industry. So, uh, before beginning, I would like to ask some of the participants, those especially those who are new to this platform, if they would like to say something. Uh, I think Arunima Ghosh, Dhirendra Dayat, are you there? Arunima? Anybody else who have joined the round table discussion for the first time? Sarvesh, Sairi Singha. See, this is a platform to interact with each other, and I would encourage those who are first timer, or at least those who are at a student level, because otherwise we may discuss something that you may not catch up later on. So we would like to hear from you first. Ishika Yadav, very nice to know that you are new to this platform. Ishika, what are you? Yes, Realme. I think your name is not there, so your name is coming as Realme. So thank you for joining. So you both are new. I would like Ishika to say something about uh, your background. Are you a student or a faculty member or a researcher? Ishika, are you there? You can switch on your microphone and tell us maybe just two, three lines. Yes, Ishika.
Yes, sir. Yes, sir. Hello,
Mhm.
sir. I am
Thank you.
Ishika. I am though I am from
Uh,
Sri Shankar.
Ishika, I think your voice is breaking. Uh,
And I am a student of my
Hello, Ishika,
Mhm.
are you done? I think
Yes,
your
sir.
voice.
Hello.
Yes,
Hello.
your voice was
Sir.
infrequent. So far, I understand you are a BTech
Okay, sir.
biotech student.
Okay, sir.
Now your voice is coming clear. Yes, tell me. You are a
Okay, sir.
BTech biotech student, and
Sir,
which
I
year?
am microbiology student.
Microbiology. Okay,
Yes, sir. I am doing MSc microbiology.
MSc in microbiology.
Yes, sir.
Great. So then you must have heard of all the enzymes that bacteria or even fungal system produces. You must be aware
Yes,
of
sir.
amylases, proteases, even
Yes,
cellulases,
sir.
right?
Yes, sir.
Uh, and to some extent their production system. How can we produce it when there are different organisms that can produce such enzymes in large quantities, right?
Yes, sir.
The most uh, common application in every household is the use of detergent, where these
Yes, sir.
enzymes like cellulase or amylase or proteases are mixed or immobilized on a support matrix, and with your detergent these enzymes are provided to each and every packet of the detergent powder or liquid detergent that we purchase, right? So
Yes, sir.
even every household is using such enzymes, right? So glad to know about you, Ishika. uh, Next, Shreya, I guess, Sairi Singha, you are also new. Please tell us about yourself and your background.
Good evening, everyone. My name is Shairi Singho. I'm an MSc Microbiology student from Kolkata.
Okay, so glad to know that you are also microbiology student, and it is very important that when we will start discussing the topic, we can have some questions from you all. Glad to know about you, Shairi.
Thank you, sir.
Jyotsna, I think, na, Realme is Jyotsna or Joshna is the name. Are you there, Joshna? Okay, anyways, okay, so we can start the topic, and uh, to all the microbiologists available to the platform, I hope they already know. Those who don't know, I would like to tell that there are microbes, especially fungus, which can produce a lot of enzymes that are of industrial importance. And when we talk about industrially important enzymes, there are we can just consider the term enzyme itself. So enzyme have some classification number or EC number. So this means they can conduct biological reaction. They are biocatalyst. They don't get consumed during the reaction, but they convert some product, some substrate to product, right? And they can be reused. So this is the important feature that enzyme have, that they are not being consumed during the reaction and they can be reused. So once if they are immobilized, so these enzymes can continue the reaction, although the over a period of time they lose some of the efficiency, but still at least for 7 to 10 cycles we can use the immobilized enzymes. Now if we talk about the different types of enzymes, I have already said amylases, proteases, lipases are there. Then there are hydrolases, cellulase, pectinase, lactase. So there are different types of enzymes that are of industrial importance. Nowadays, whenever we talk about 2G ethanol or biorefinery or waste conversion to a value-added product, we need these fungal enzymes, especially hydrolytic enzymes that can degrade cellulose or convert lignin or degrade some amount of lignin so that cellulose gets free for the next step. So all these things are possible at a large scale only if we can use these enzymes properly. And whenever we talk about production of enzymes, it is not that we have to produce them only in flask. Okay, so we need a very strong organism which can produce it using cheapest carbon source, as cheaper as, like, maybe just yeast extract, or maybe just minimum salt minimal salt media with some basic components, and that organism can grow and produce the enzymes. So enzyme production is again condition dependent, temperature dependent, pH dependent, right? So all these things happens uh, because of the property of the strain, and the strain property can be developed or enhanced through mutagenesis or through various other approaches, right? If you talk about fungus, So fungus will have a specific fungus will have a specific system like uh, if you are using solid state fermentation, you can produce such enzymes very cheaply. But not all the enzymes will uh, be produced in solid state fermentation. So there are various aspects of enzyme production. Now, having this as a background, and some of the industries like uh, if we talk about some kind of reaction, specific reaction or amination, transaminase reaction, transesterification reaction. In those cases, enzymes such as lipases gets involved, so they are used a lot in all these cases. Right. So having this in the background, I would like to open the discussion forum and would like to know from the participant if they have been working on such enzymes uh, in recent time. Okay, so the stage is open for discussion. I would like to call upon Anusuya Das. Anusuya Das, are you available? Anusuya, Arunima Ghosh, Chanchal Kumari, Dhirendra. So anybody who would like to speak or they are working on fungal any type of enzymes, or if they would like to in future they would like to work on enzymes, so which type of enzyme you will you work? So those who are especially microbiology background or student level, they can discuss more if they would like to continue their work on enzymes. Gati Patel is also new, I think.
Good
Gati
evening,
Patel,
sir.
are you there? Yes, yes,
Uh,
Tejas.
sir, I am
Tell me.
audible.
Yes, yes, Josna.
Yes, yes, sir. Sir,
Yes,
my
tell me.
work presently my work is going under the Aspergillus niger and Aspergillus fumigatus. The both are the
Mhm.
produce enzymes like cellulase.
Hmm.
Sir, how it is confirmed these are the generally the research paper research background I know that these are the produce cellulase. So how it is confirmed these are the produce cellulase enzyme.
Okay. So, very good question. And this is a very, very important topic whenever nowadays you may have been hearing about the Sustainable Development Goal as well as the utilization of waste to value-added product or biorefinery. Okay. And whenever you are using a kind of biomass that is agro
Sir,
based, so they will,
I have already used to the different type of the biomass like corn, banana stalk, potato,
yes,
rice,
yes,
etc. Then after that the like my total whole topic is bioethanol production. That's why the one of the part is enzymatic hydrolysis. I have just extract the enzyme, the different microbes. So generally my students are used to the enzymes cellulase enzymes are the produced from the Aspergillus niger and Aspergillus fumigatus. So, sir,
yes.
generally, how it is confirmed? The student, I think the some of the students are there. So, kindly just confirm and explain that the matter.
Yes, the very simple. Yes, I was coming to your question. Tejas, you want to say something?
Yeah, actually, I'm also working on the similar topic, so I was just sharing my views. But it's okay if I can also answer the question, but you can continue, sir. No problem.
Okay. So
Sir,
I was coming to the yes. So I was coming to the answer, but to other audience, those who may not know. So I need to give them a little bit of background that if we are using any kind of agricultural residues, they will contain lignin, hemicellulose as well as cellulose as the biomass. So out of that, cellulose is the most utilizable material because it can be degraded to release. glucose molecules, and that glucose can be converted into, in your case, it is ethanol, but it can be converted to any other byproduct, right? And so here our target is to first to get cellulose. Okay, and then if you get cellulose, the enzyme that you are using must be able to degrade it. So by this, having this knowledge itself, you can get the idea that the assay that you have to carry out is to use cellulose as a substrate in a buffer and put the enzyme to work. And if the glucose is released, estimate the amount of glucose. Okay, so for glucose estimation, typical reaction is DNSA method. So you can use DNSA method to check the level of glucose, right? So that is one of the reaction. Universal standard or global standard is mostly followed at as NREL. You may write it down, those who are actually working on it. So NREL protocols are there for all such work on biomass to bio or bioconversion reactions. Okay, so there you have one protocol, standard protocol of estimating cellulose, as well as how to check the cellulolytic activity of any enzymes. Another one is the method of Ghosh et al., which is very famous and highly cited. Each and every paper you may read even nowadays will be citing Ghosh et al. For its FPU activity or assay method. FPU is filter paper activity, and FPU, if you are counting as FPU, it is filter paper unit. Okay. So filter paper assay means you will use filter papers that are made up purely of almost purely of cellulose. So you will use filter paper as a substrate, give the enzyme, use citrate buffer point, I think 50 millimolar citrate buffer you need to use, pH 5 to 6. That depends on the enzyme from where you are getting, and then allow it to work for some time. I think it is 1 hour you allow for the reaction. Then you stop the reaction and check for the released sugar through DNSA method. So the amount of sugar released is directly proportional to the amount of filter paper degraded. Right. So that is a standard method by Ghosh et al. You can search it online also. I hope I was clear,
Okay, sir. Thank you so much.
Josna. Yes, yes.
Okay.
So you can ask your students to perform DNSA method. It is very easy. 5 minutes boiling with DNSA reagent. Uh,
No, sir.
right.
DNS method.
So after you allow the filter paper to degrade, then go for boiling, cool it down, and then calculate the amount of sugar released. Yes, Preeti Rani, you want to say something?
I would like to add one thing to the same
Yes.
question that was asked. Yeah, so if your enzymes are produced, I mean, I'm also working on the similar enzymes like cellulase, xylanase, and lactase. So if your enzymes are produced in the SSF, then you can simply check whether the produced enzyme is cellulase or xylanase or lactase or any enzyme by simple plate method. So you just have to take zero point 0.5 gram of agarose in say 10 ml of distilled water, and you can add the carbon source that you are using in the specific enzyme. Like for cellulase, you can simply use CMC plus agarose in the distilled water. You can heat it at 50 degrees Celsius, and you can simply pour it in the petri plate. And in the well diffusion method, if you are produced crude enzyme, if you are putting the crude enzyme in the well, then you can simply observe the zone of clearance after adding say grams iodine or we can say any dye. So that can be preliminary check whether the produced enzyme in your SSF or SMF any method. So it is the same enzyme that you are working on, or else the quantitative assay it will go in vain. So I would like to add that thing only.
Yes, very right, very right. You are for a screening method. If you are screening for these kind of fungus which can produce such enzymes, then agar plate-based methods are very nice. And even for
The,
qualitative analysis, yes, qualitative analysis,
just for
it is very
qualitative
nice.
analysis of enzyme,
Yes.
I'm not talking about fungus. I'm just talking about the produced enzyme. So if
Hmm.
it's produced and you don't know whether the production or your crude substrate contains cellulase or xylanase. Sometimes the fungi which is growing on lignocellulitic substrate, so it produces many types of enzymes. Whichever substrate is easily available for the fungi to degrade, so it produces that enzyme. So just in order to check whether the produced enzyme or whether the crude enzyme contains your enzyme of interest, so you can just simply add the substrate to the agarose, so that we can just carry out the enzyme substrate reaction in the petri dish, and you can simply observe the zone of clearance on the plate after say 5 to 6 hours.
Yes, yes. And how about lacase? Have you ever tested lacase on agar plate?
Yes, sir. I have also checked lacase on the agar plate. So it similarly it shows the we can say reddish brownish zone around the well, like it shows in the screening method. So it also shows the oxidation of glycol on the plate if you are adding a significant amount of enzyme in the well. So it degrades the enzyme on the plate also. So it shows a reddish or brownish zone.
Okay, so we don't have to develop it just like we develop using iodine in other cases.
Yeah.
How
So
we need
actually,
to develop it, or it will convert the color?
yes, the quantitative method is obviously we must have to go for quantitative method. But before going for quantitative method, or say we can say before going for purification of the specific enzyme, so we can simply check whether the produced enzyme is this or that. Okay, so you can save your time as well as your resources before going, because the quantitative method for lacase, say we need ABTS, and that is I think 12,500 for 1 gram. We can simply check whether the produced enzyme is lacase, so that we can go for the quantitative method.
Yes, yes, yes. Right.
So like I'm talking now, so I can share my experience. So I'm a PhD research scholar from the process of PET 2023. I'm registered in Kavitri Bainabhai Chaudhary, North Maharashtra University, Jalgaon, and I'm working on the production of cellulase, xylanase, and lacase. I'm working on the mixture of these enzymes, like enzyme consortium for the valorization of lignocellulose biomass. And recently, in the project dissertation that was under me, we utilized this enzyme consortium to valorize the groundnut peels, so that it can be used as an enhanced chicken feed. So we fed the valorized groundnut peels to the chickens, and to
Mhm.
our surprise, after 1 week, we estimated the protein from their fecal samples, and our test chicken that was intaking the valorized groundnut peel. So to our surprise, the protein content in the fecal matter of our test chicken was say 30 to 40% less as compared to the control. So we concluded that if we degrade the cellulose, or we can say hemicellulose outside the body, and then we fed the directly ready to eat sugar to the chicken, so it can utilize more amount of proteins, and less amount of protein get discarded in the fecal matter.
Very nice, very nice work.
I have recently published article on cellulase, on the production, purification, characterization, optimization of cellulase-free xylanase from a fungal isolate that I have isolated in my laboratory. So it got published
Okay.
in the International Journal of Biological Micromolecules with elsewhere, and we also enhanced the enzyme activity using ultrasonication, that is a physical method. So that is also a very we can say upcoming topic in the field of enzymes, where we can increase the activity as well as increase the production of the enzymes using physical methods like ultrasonication.
So you used pure enzyme or crude enzyme
No,
for
actually,
ultrasonification?
the paper included firstly the isolation of new strain, that was Pineophora incarnata. We have deposited that strain in the NCBI, So we produced xylanase enzyme from Piniophora incarnata. Then we purified the enzyme, we characterized the enzyme, and then at the end we checked the ultrasound assisted activity enhancement. So the activity was say 1.5 fold increased after the ultrasonic treatment of purified enzyme.
Okay.
Yeah.
And one more thing I wanted to ask is that in your work you used consortia. So it was consortia of fungus or consortia of bacteria or
No, no.
both?
Actually, my PhD work is focused on the consortia of enzymes, but while isolation,
Consortia of enzymes of means purified enzymes or
no, we can use partially purified enzyme because, as I told
okay.
you, means I am actually working on this in the lab level. So, uh, any fungus that is producing say cellulase, so it ultimately produces lacase because it is growing on the mixture of lignocellulose. So, okay.
Yes, yes, yes.
uh, in order to get the cellulose, the fungus first needs to produce lacase in order to break the lignin layer. So my main focus is production of these three enzymes from a single strain, or we can say multiple strains, and then partially purify those enzymes, and then we can produce a mixture. We can optimize the mixture. We can say produce a formulation that can be used for the valorization of specific type of lignocellulosic biomass.
Hmm.
So
Okay,
I
very, very.
got
Uh,
many applications which are currently under publication. But yeah, it actually opens up a very huge path for industrial enzymes. So, like you told in the introduction part that many enzymes are used in the industries. So, by using these type of mixtures, we can simply reduce the time that is required for the breakdown of any substrate. So, especially the complex substrate like lignocellulose, which have multiple polymers mixtures in the substrate itself.
right, right. Great. Thank you, Tejas, for your useful insights. And I think you may have seen the chat box. Miss Josna would like to connect with you. So if you can just share your mail ID in this chat box. So it will be helpful for us and other participants as well if they want to get in touch with you.
Okay, sir, fine. I share my.
Yeah, yeah.
Actually, I'm
So
also
very nice work.
PhD student. I'm currently working on it, but I just shared my experience because MBSI has granted me the PhD fellowship recently. So since then,
Okay, very
I just
congratulations.
yeah,
Hmm.
I appreciate this effort. I mean, multiple researchers come to a platform and they discuss their research topics. So we can also get new idea in our own research topics. So it's a great initiative. So that is why I join each and every meeting of yours.
Yes, yes, yes. Yes, so with the participants like you only, this discussion forum will become more and more interactive as well as useful to for the topic specific means those who want to get some information. So it is very nice
Yeah.
that you talked today, and we are glad that you are also engaged in all the meeting. Yes, the certificate will be emailed to you. I will. I think who is asking. Jagatia Kirti. So I will share the feedback form after some time, maybe in 10 minutes. So that feedback form you will fill and you will get the certificate via email. Okay. So you. How about Jagati? Are you there? Yes. If you would like to tell something about your work or your background, you can share with us. Jagatia, you have to unmute yourself if you are available. You can unmute yourself and then speak. So, in continuation with what Tejas has said, there is one unique application that I also got to know about that was unique because it works as a food additive or kind of helpful for the gut. So, we need some of the oligosaccharides for better digestion, and so there are xylo-oligosaccharides which are much in demand nowadays. So xylo-oligosaccharides are also produced by unique type of enzyme that can only produce xylo-oligosaccharides, not other monomers. So by having such enzymes, so basically they will cut or chop the molecules, semicellulosic cellulose molecules, so that you will get very shorter uh, or basically oligosaccharides of xylose that are helpful for the gut, and it is medically very important for gut related diseases.
We are also heading ahead, continuing the project. So we have also came to know that if we give the oligosaccharide directly to the chicken, so the energy that is it is utilizing in breaking down the polymers, so that can be utilized in gaining the weight or say laying the eggs. So we can also increase the yield by just by feeding it the enhanced feed. So
Mhm.
we are heading towards that. But in the preliminary test, we just got to know the protein estimation of its fecal matter. But we just read that this can also be done. So it is also a forward application of enzymes.
Yes, and we have Professor Prakash Halami sir, who is much knowledgeable in this aspect, especially gut displaces and oligosaccharides and all. So, if, Prakash sir, are you available today?
Yeah, I'm very much available, but my network is little bit poor. So if
Okay,
any
okay.
specific questions, I can actually answer by by email subsequently.
Okay, okay. Okay, sir, no problem. Prakash sir is have been working on various aspect of microbes, including probiotic bacteria, lactobacillus, and all. And while I am talking about lactobacillus, there is another enzyme that is degrading lactose. So people are also trying to make formulations to degrade lactose in the
Also,
milk so that
molecular biology, the restriction enzymes, right?
yes, yes. That aspect we have not yet covered. Yes, sir. Very nice point. So restriction enzymes are used a lot in all the molecular biology lab. Whatever we read, even the CRISPR-Cas9 system is also a kind of endonuclease, but very specific endonuclease that has led to a complete transformation of how genetic engineering is happening nowadays in the lab. So I hope some of you may be working on molecular biology aspect or maybe using restriction enzymes or maybe CRISPR. So if someone is working on such enzymes, they may also come forward and discuss. Today we have very shall we call upon the participant or if anyone of you are working Bharat, I saw Bharat, he left, I guess. So Yogesh, Smita, Shraddha, are you present? Would you like to say something? So, dear participants, I think we have much of the discussion. Arif, would you like to say something about your background or your interest in the topic? Arif, Anusuya, if you are hesitant asking question, you may also drop your questions in the chat box. As Professor Halami sir has said that he may respond to your questions via email as well. So you can drop your questions as well as your email ID so that we will address your query. While I would like to encourage all of you to ask questions and discuss. Uh, Chanchal, Jagatia, Kavita Tambe. So, anyways, I think uh, we had quite a bit of discussion, and now participants are already engaged, it seems. So, I have dropped feedback form for you all. Those who are present must notice that they need to fill this feedback form. I think some Kriti, Jagatiya Kriti, you were asking about the form, so I have sent the feedback form. You are requested to fill it so that you will get the participation certificate. And I would like and request all of you to join the next session of MBSI roundtable meeting, and the topic will be sent to you over our WhatsApp group so that you will be getting notified about the event. So if there is no more questions, uh, we may stop our discussion here. But before stopping, I would again like to ask all of you, if anyone having any question or they want to discuss something, they can come forward and discuss. Yes, Anusuya Das. Anusuya Das, you'd like to say something? Dr. Gopalji Gopal is also available today. Dr. Gopal sir.
Just joined. I missed
Yes,
lecture.
sir.
That
I noticed you. No, still it is fine, sir. I was about to end, but since you are available, we would like to have your useful view on the today's topic, which is on industrially important enzymes, microbial enzymes.
so have you talked about recombinant enzyme or just from
Uh,
natural enzyme using?
as so far, only natural one, and recently Prakash sir also noticed that I am missing the restriction endonucleases or restriction enzymes, which are a lot means which are used in huge quantities in all the molecular biology lab.
Yes. Nowadays, all enzymes are produced through recombinant DNA technology because production is more, है ना?
Mm.
Because we clone it in a strong promoter. So from natural resources, we cannot control that strength of the promoter because it is present in their natural promoter.
Yes, yes, yes. So all this recombinant DNA tech comes under the strain development program, where either through natural selection or through random mutagenesis or via controlled changes in gene expression using recombinant DNA technology, we can improve the strain. Right,
Yes.
sir?
Means if gene is known, gene sequence is known, then we should prefer to clone a new host, है ना? E. coli host, or if there is post-translational modification required, then higher organism, है ना? Like yeast and other. So that we can produce more amount of protein using E. coli host, mostly.
Yes, sir.
What is preferred? Natural, from natural sources, means native host or new host? What is your suggestion?
No, for industrial scale, I think recombinant host is always better because we can fine tune the yield or production of the enzymes.
Yes,
We can
yes,
control the
yes.
expression through the promoters.
Yes.
So, but sometimes or rarely we find a very good strain, but that is quite rare. While here we can develop, if we know the gene, we can develop it, develop the strain itself. So
Yes.
yes,
Sometimes if gene is not known and that organism is complex, newly discovered, suppose some extremophiles, then we can go and culture that organism and then we can produce from native host. है ना?
yes, sir, correct.
I could not join on time. Today was
No,
Tuesday.
sir. Next time I will be waiting for you. I will also message you. Please send your email ID.
Yes,
Because
yes, yes.
so far I remember even I sometimes miss the meeting due to previous engagements or so. But uh, uh, the last time we had a discussion, it was quite fruitful, and all the participants like that meeting as well. So
Yes, yes, yes.
yes, sir.
Acha, Microbiology Society is very active society.
Yes, sir. We are
Always
trying
doing
to
something, है
be.
ना? For
Yes, yes.
dissemination
Today.
of knowledge.
Yes, yes, sir. Today, Mr. Tejas was very actively participating. He is awarded as a PhD fellowship in this through Microbiology Society of India, and
Okay.
he started participating and is actively engaged also. So today he talked about his work, which was quite nice and unique. Thank you so
Means
much,
I am
sir,
attached,
for
I am
joining.
associated with several organization, but I have seen कि Microbiology Society, they are actively, है ना, engaging scholars and faculties and students, trying to disseminate the knowledge. है ना? There are
Yes,
several
sir.
WhatsApp group, and in WhatsApp group also people are exchanging their knowledge. Other society, they just conduct one annual conference seminar and they think they have completed their job. है ना?
Yes, yes, yes. And it is all because of the initiative taken by our President, sir, Professor Arvind, sir, and from the participant life members also, sir, like you, Prakash Alami, sir, Vipran, sir, and so many others who are engaged in different activities across the year.
Yes, yes.
Even
बहुत अच्छा.
I have started the student unit, although I could not make much of the events. Only two events I could conduct so far, but slowly
That
I
is
got.
also good. Two event is also good. If every student unit will start doing two at least annually, then it also becomes some more than 100. है ना?
Yes, sir. As a
Mhm.
microbiologist, we especially celebrate a International Microorganism Day.
Yes, in September, na?
Yes, sir.
Yes, it's good.
Mm. So thank you so much, sir, for joining. Although I think we would like to wind it up because
Thank you. Yes.
are there for the almost for the past 1 hour. So with this, I would like to conclude our today's meeting, and I would like to thank all the participants who spent their time for today's roundtable discussion. I would like much of the students should join, so those students who have joined today may ask your connections to come forward and join MBSI as well as this platform to get more and more benefit. And thank you, Tejas, Josna, and other participants who have been actively engaged today.
Thank you so much, sir. Thank you for the opportunity.
Yes, yes. Dr. Parakram is there. Hello, Dr. Parakram. Parakram Singh Chauhan, would you like to say something? Okay, then I would conclude the meeting. Thank you, all the participants. We will join again in the next week meeting. Thank you so much. And before leaving the meeting, do fill the feedback form. Okay. Feedback form is sent again in the chat box.